So this video, the topic is about ELISA, Enzyme-linked immunosorbent assay. Enzyme-linked immunosorbent assay, also called as EIA, enzyme immunoassay. The test to detect and measures antibodies in the blood sample. So it may detect antibodies of HIV, hepatitis, or many more infectious agents. So why it is called ELISA, enzyme-linked immunosorbent assay? Why it's named as ELISA? Since the name ELISA is using the sorbent, that means adsorbent. Antigen or antibody are linked or adsorbed with the plastic surface. That's why it's called sorbent. And antigen is recognized by specific antibody. There will be an immunological reaction. That's why immunosorbent. Then third one, the test utilizing an enzyme linked with secondary antibody for detecting the presence of primary antibody with the help of substrate. That enzyme substrate reaction gives the colored product in the end of the test, at the end of the ELISA test. So that's why enzyme linked immunosorbent assay. So we are calling ELISA since the enzyme mixed with the substrate is used to detect the presence of antibodies of interest or antigen of interest. So these are the basic terms involved in the ELISA test: solid phase, adsorption, washing, antigen, antibodies, enzyme conjugate, chromogen, stopping, and reading. So these are the basic terms used during the ELISA test. Solid phase means the microtiter plate which is used to detect the ELISA, detect the antibodies in the ELISA test. So this solid plate, that is plastic plate having A2:12 well format. Adsorption, that is the process of attachment of antigen or antibody with the solid phase, with the help of buffer during the incubation. So during the incubation period, the antigen or antibody of interest is attached with the solid support, that is a microtiter plate. Washing, this is the process usually done each and every step of the ELISA test. So washing can be done with the buffer, right? Then another is the antigen and antibodies. Ultimately, antigen is a molecule that elicits the production of antibodies. So it is normally present in the patient sample, and the antibodies also present in the patient sample, right? These are the proteins in response to the antigenic stimuli. So the antibodies will be produced in the response to the antigen. So antigen will stimulate the production of antibody. Then enzyme conjugate. That means there is an enzyme, for example, horse Serology peroxidase will conjugate or attach with the secondary antibody. It's called as enzyme conjugate. Chromogen is a chemical substances like trimethylbenzidine. This chromogen will react with enzyme, will give the color product, that is the chromogen. Stopping. So stopping buffer is normally used, or stopping chemical, it is normally used for stopping the reaction, stopping the enzymatic reaction during the ELISA test. Reading, this is the end of the process. The reading of ELISA test can be done spectrophotometrically. So that is the reading. So next is the principle involved in the ELISA. So the basic principle involved in the ELISA is about the antigen and antibody reaction. Antigen and antibody reaction. For example, in our normal body, when the bacteria or any virus or any infection enter into the body, that infection will be considered as an antigen in our body. So our body will consider particular infection as an antigen. So once enter inside the body, that antigen will stimulate the production of antibody in the patient body. So that antibodies will react or attach particularly with the specific antigen. So the antibody produced by the human will fight against the particular antigen. That is a normal physiological process or immunological process happening in the normal human beings. So that is what happening in this case of ELISA test. So here antigen is present in the microtiter plate. When you add the patient sample containing antibody, that antibody will attach with the antigen. So this is the basic principle involved in the ELISA. For example, the patient having HIV, the patient sample having the HIV means the HIV antigen is coated in the microtiter plate. When you add the sample in the microtiter plate, if the patient having the HIV antibody, then there will be an antigen and antibody binding. That is what we are detecting in the ELISA. So the detection can be confirmed by enzymatic reaction. That gives the color product. If the antibody present in the patient sample, that gives color product. If no, there is no color product. So that is what happening in the ELISA. So this antigen antibody reaction can be compared with the lock and key concept. So the particular lock can be opened only by the key, only by the specific key, particular key. The same thing will be happening in this case of antigen antibody reaction. Particular antigen can bind only with the specific antibody. So it is very particular about the binding and it is very specific about the binding. So that antigen antibody binding will make the detection of any antibody presence in the sample. So that ELISA test will make the detection of HIV present in the patient sample. So this is the principle involved in the ELISA. So what are the reagents used in the ELISA? So the reagents are some puffers we are using, right? Like coating puffer, washing puffer and blocking puffer. These are the three puffers using in the reagent of ELISA kit. So another thing is the enzyme and chromogenic substrate and stop solution. Puffers include, the composition of puffers include phosphate puffer, NaCl, that is coating puffer, phosphate plus NaCl plus 220, that is a washing puffer. Blocking puffer or blocking agent is the bovine serum albumin. Enzyme normally horse aradis peroxidase. Chromogenic substrate is the trimethyl benzidine. Stopping solution is 0.5 m H2SO4. So this is the general procedure, the slide showing the general procedure. So the ELISA test having the 5 steps. First is the antigen attachment with the plate, microtiter plate. So in the kit, ELISA kit, in the ready-made kit, antigen is attached or immobilized or conjugated with the microtiter plate. Not only antigen, you may attach the antibodies also. So if you want to find out the presence of antigen, you may attach the antibody. If you want to find out the antibodies present in the sample, you may attach the antigen. So in this picture, there will be attachment of antigen. So there will be an attachment of antigen in the microtiter plate, right? So after this, you have to add the blocking buffer. So blocking buffer will block unnecessary antigen binding sites. So that allows the primary antibody will bind specifically with the particular antigen binding site. So blocking buffer will be ordered. That is the second step. The third step is that addition of suitable antibody. That is a suitable antibody means that is the antibody present in the patient sample. So after adding, primary antibody will bind with the antigen. There will be a formation of antigen antibody complex. Secondly, there will be an addition of secondary antibody. That means anti-antibody conjugate, enzyme conjugate. So after adding this secondary antibody, enzyme conjugate, secondary antibody will bind with the primary antibody, FC portion. Then last one, addition of substrate. That substrate will react with the enzyme that gives the color product. This is happening in the ELISA. So this picture also showing the same, but this picture showing the biotin, streptovidin, horse heredis peroxidase reaction. So here the antibody is attached. So the first picture, the antigen is attached with the microtiter plate. Here antibody is attached with the microtiter plate. Here we are adding the antigen. So here there will be a antigen and antibody complex. Then we are adding the enzyme conjugates such as biotin, streptovidin, then horse heredis peroxidase. Then we are adding the substrate. There will be a colored product at the end of the ELISA test. Then we are going to discuss about the types of ELISA test. So there is a 3 types of ELISA test: indirect ELISA, sandwich ELISA, and competitive ELISA. Indirect ELISA, we are detecting the antibodies, presence of antibodies in the patient sample indirectly, since we are attaching the antigens in the microtiter plate. So there is a antigens attached or coated with the well, then primary antibodies attached, there will be a complex formation, then secondary antibody containing enzyme conjugate, there will be a color product. This is the indirect ELISA. Sandwich ELISA means we are using the antibodies coated with the microtiter plate. So here there will be a antibodies coated with well, then there is an addition of antigen, there is a complex, then followed by there is a enzyme secondary antibody complex is attached with the antigen, then there will be a product after addition of substrate. So this reactions is happening, or attachment is happening like a sandwich, one above another, looks like a sandwich. That's why we are calling it as a sandwich ELISA. Competitive ELISA in the sense, in the microtiter vessel, there will be a competition between the antibodies for binding sites present in the antigen. Here we are mixing the 2 kind of antigen at the same time. That means primary antibody and secondary antibody. There will be a competition between primary and secondary antibody to bind with the antigen. So here the primary and secondary antibody will started making the competition or fight for binding with the antigen. So that's why we are calling it as a competitive ELISA. So in this case, the bound antibody and unbound antibody competition will be there, right? After washing, the unbound antibody will be taken away from the vessel, right? Only bound antibodies will be present in the vessel. That gives the color product. That is the competitive ELISA. Next is the advantages of ELISA. Main advantages is cheap and it has a long shelf life. This ELISA test kit having the long shelf life. It is also somewhat cheaper when as compared to the other tests. For example, most of the diagnostic tests need a sophisticated instrument, but ELISA just the test and we may be able to detect spectrophotometrically. Moreover, it's highly specific and sensitive. as compared to the other diagnostic tests. So specific and sensitive. Then there is no radiation hazard since we are using enzyme as a enzyme conjugate for detecting the antigen or antibody. So there is no radiation hazard. We are not using any radioisotopes like radioimmunoassay. So there is no radiation hazard. It's easy to perform and quick procedure. Yes, quick procedure. Equipments are widely available. Then there will be a chance of detecting variety of infections. Such as HIV, hepatitis B, hepatitis C, and syphilis, chlamydia infection, etc. We can detect many kind of infections. And moreover, it is used most type of biological samples. So we may able to use plasma, serum, urine, or any kind of cell extract in order to detect any diseases or infections, or we may detect any kind of serum or protein with the help of ELISA test. Disadvantages that includes measurement of enzymatic activity is more complex than the measurement of activity of some type of radioisotopes. When compared to the radioisotopes, this enzymatic activity is somewhat complex in nature. That is one of the disadvantages. Moreover, this enzymatic activity may affect with plasma constituents present in the patient sample. So some constituents present in the blood may interfere with the test results due to the enzymatic activity of the enzyme conjugate. Then kit is not that much cheaper, so comparatively somewhat costly when compared to the other cheaper kit. Moreover, it's very specific and particular antigen, specific to the particular antigen. It won't recognize other antigen. So actually it is not the disadvantage exactly. But whenever we want to detect, whenever we want to go for other antigen detecting, we may consider as a disadvantages. For example, if you want to detect HIV, we must have a kit for detecting the HIV. So the kit must have the antigen, HIV antigen coated kit. So that kit only useful for detecting only the HIV. We cannot able to detect the other bacterial infection or other viral infection with that same kit. We have to look for the separate kit. Then there may be some false positive and negative reactions due to the altered or mutated antigens. In some cases, for example, HIV patient may have a antigenic shift or mutation of antigen. So that time there may be a possibility of negative results. Applications of ELISA. Applications of ELISA includes the screening of any viral contamination during the blood donation or blood transfusion. For example, we may able to screen for HIV or hepatitis during the blood transfusion. This is a normal process takes place during the blood transfusion or before transfuse any blood into the another. So this is the normal screening. So it can be done with the ELISA. Then measurement of hormonal levels. So with the measurement of hormonal levels, we can able to diagnose the many hormonal disorders. We can go for treatment with the help of this measurement of hormonal level, such as human chorionic gonadotrophic hormone. We can able to detect the pregnancy exactly with the help of measuring the human chorionic gonadotrophic hormone. Then luteinizing hormone, thyroid stimulating hormone, we can able to use this measurement of hormonal level. Then detecting infections, we may able to detect variety of infections such as HIV, hepatitis B, C, syphilis, chlamydia, etc. Then we can able to detect some illicit drugs. We may able to detect the concentration of some illicit drugs, and also we can able to detect some allergens present in the food or any other samples. Thank you.